And I'd like to introduce Walter Wynn to take it away and introduce our program for today. Thank you.
Good morning. We are really doing learning at a distance today, but I will let Andrea introduce our speaker. I'm listed as the facilitator, but all I do is make this short announcement and turn it over to my excellent recruiter, Andrea Cunningham, who spoke to us a couple of years ago. She actually has located all of our speakers and will introduce them. Andrea, you're on.
Thank you, Walter. So our speaker today, I have the pleasure of introducing Rose Bryson, who comes from Anchorage, Alaska, and she's currently there right now. And she received her Master's of Science in Bioarchaeology and Forensic Anthropology degree from the University College of London in 2016, and is now a PhD student in the UF Department of Anthropology. Her dissertation topic studies how the skull changes as you grow and develop, specifically how the growth of the brain and the eye affect what the adult skull will look like. And she's hoping to use this information about the anatomy to create an age estimation technique for forensic identification efforts on skeletal remains. Rose currently works as an analyst at the CA Pound Human Identification Lab in Gainesville, where she responds to crime scenes, analyzes skeletal remains, and creates 3D digital models of bones. And after she receives her PhD in 2023, Rose hopes to serve as a forensic anthropologist working with law enforcement to identify skeletal remains. Rose, we so appreciate you being here today, and we're really looking forward to hearing more about your research. So you can start your presentation whenever you're ready.
Sounds good. Good morning. Thank you so much for having me today. I love talking about bones and forensics, so I'm excited to talk this morning. All right, let me share my slides and we'll get going. There we go. And I am lecturing from a distance this morning. I'm in Anchorage with my family. So I want to give everyone in the audience just a warning before I begin so that you can be prepared. There are photos of actual human remains in this presentation. I don't show anything too graphic. I'm not here for shock value, but I do have pictures of real bones, real skeletons. And I know there are some biologists and anatomists in the audience, so you're probably used to a lot of this. But the photos are either from public textbooks or people who have donated their bodies with permission to be used in teaching and research to a forensic anthropology facility. So I was given permission to use them in today's presentations, but if you would please not take any photos or screenshots, that would be great since these are human remains. But I'm going to give a brief description of just what forensic anthropology is, and then I'll introduce myself a little bit and we'll dive into just exactly what you can learn from a person and everything you can learn about pieces of people's life story when all that is remaining is their bones. So let's move on. Okay, so today I'm talking about my field work, which is forensic anthropology, the study of human skeletal remains in a medical legal context. So this means that I work as a specialist of the human skeleton and consult for law enforcement or medical examiners when they find skeletonized remains. So anthropologists really study everything having to do with humans and human behavior. So we are anthropologists in a forensic world. And so in that world, we're really trained to excavate a crime scene very slowly and very carefully, the same way you would excavate an archaeological site. And that really helps us to retain as much evidence as possible. So we're usually at this perfect balance between, law enforcement wants us to go really fast and get the job done, and archaeologists really want to take their time and get every single speck of evidence. So it makes a nice balance that we're collecting everything really important, mapping things in, surveying very carefully, but then also have some real world constraints of get the job done and collect everything. So sometimes our job means hikers find bones in the woods or on the beach or in the mountains, and they think that they're human, so they send them to our lab. And most often we have to disappoint people and say, sorry, these are, or not sorry, but these are deer or raccoon or bear. It's not a human. But other times we're called to a crime scene to search or recover remains of people who are missing or have been murdered. So our job as forensic anthropologists is to put together these last clues of who someone was, and try to link our estimation of their sex, their age, how long they've been dead, with a missing persons report to help complete that story of what's happened to somebody after they've gone missing. So Andrea already gave me an intro, but I'll just say I wanted to, before we dive into content today, just quickly introduce myself. My name is Rose Bryson, and I put my email at the bottom here. If you have any questions after today, please reach out. I love talking about bones and forensic anthro, so feel free. I can also put it in the chat later on, but feel free to reach out if you think of anything you want to ask later on. But I'm currently in my 4th year of my PhD at University of Florida, and I work at the C.A. Pound Human Identification Lab, which is a forensic anthropology lab in Gainesville. And I've known since high school that forensic anthropology was a career that I wanted to pursue. So I chose to go all the way from Alaska to University of Tennessee, specifically so I could work at their forensic anthropology center and learn how to do this sort of forensic work. And then after that, I got my master's in University College of London. And now I'm in Florida, working at the forensic lab and trying to get my PhD so that I can become a fully fledged forensic anthropologist. All right, so let's begin. We start collecting information about a case from the time that we step right into a crime scene. So the way that the bones are laid out or oriented, the type of environment that they're in, all these can be really important clues to determine what's happened to that person. So we typically collect the remains from a crime scene or are sent them by a medical examiner, and we produce an osteological report. So osteo just refers to bone. So it's a report on all the information that a skeleton can give us, and then we give that back to the investigators. So that report is going to include a biological profile. And the biological profile is an assessment to try and regain some of that information of who that person was that's lost when they die. So the 4 components that we can kind of regain from the skeleton are sex, age, ancestry, and stature. So how tall they were before they died. And by reconstructing those pieces of someone's identity through their skeleton, we can help law enforcement, investigators link these unidentified skeletal remains to a missing persons report. So we'll go through each one of these, just briefly, each one of these components of the biological profile to show just how we get this information from a skeleton. But you can see on the right, we can only provide an age range or a stature range or an estimation of sex and ancestry. We can't be as specific as a lot of TV shows would like you to believe that and say, oh, with 100% certainty, it's a 44-year-old from Oregon. Unfortunately, we can't get that specific most of the time, but we do provide these estimations and age ranges so they can say, all right, we'll exclude all these people that are missing persons reports and include these things that fit with the biological profile. So we'll just go through these different components real fast. So how do you tell age from a skeleton? There are generally 2 categories for assessing someone's age from a skeleton. There's the techniques you use when someone is a juvenile, and there's the techniques you use when someone is an adult. Because as a child, you're tracking what things are forming in the body, what things are emerging. You have teeth erupting from your baby teeth to your adult teeth. You can track, on the left here, we have a diagram of the mandible and the maxilla. So your 2 teeth dental arches, and the shaded in gray teeth, which you can also see a real example of over here on the right. But the shaded in teeth are your baby teeth, your deciduous teeth, and then the whiter teeth are the adult teeth coming in. So it's just a diagram of at certain ages, what is actually erupting in the mouth. So when you're doing juvenile age estimation, you can track, which baby teeth have been lost. You can track which adult teeth have erupted or are still forming. And you can also look at bone fusion. So there's a picture over here on the right of most of our bones of a femur. So the longest, your thigh bone, the longest and largest bone in your body. And most of our bones begin in multiple pieces in the entire body, from your head to your toes. They'll begin in multiple pieces and then fuse together through life. And different bones throughout the body fuse at different times in your life. So one bone might be finished fusing at 14 years of age. Another bone, like the clavicle, might not finish fusing together until you're 30. So when the clock stops as someone dies, we can see what stage of fusion different bones around the body are in, and we can see what stage of eruption the teeth are in, and it creates this narrow age range that we can estimate how old that person was. But then at a certain point, all your adult teeth have come in, all your bones have fused, and you're skeletally an adult. So we can't use those same techniques that we use to age juveniles with adults. So what you're actually assessing when you're trying to estimate age in an adult skeleton is really how the skeleton, the fully adult form of a skeleton, adapts and degenerates through aging. So because of that, the adult aging techniques are less accurate and the categories are more broad than juvenile aging techniques. So when in the juvenile, like we saw in this previous slide, you might be able to say, oh, this individual was 10 years plus or minus 2 years. The age range, those categories get much more broad. So you might only be able to say this person was between 35 and 60 years old. And what you're actually seeing on the screen is pubic symptoms, a surface on your pelvis that we look at to estimate aging. So this picture over here on the left is the youngest. Sorry, it says my connection is unstable. Can you still hear me?
You cut out
Yes.
for a few seconds, but we can hear you now.
Sorry, but next time I'll do a talk in person. But I appreciate you guys learning from a distance here. Your pelvis. Photo on the of youngest to oldest is look like in, say, an 18-year-old, and then track what appears, how this surface change until, say, you're 70 or 80, and that would be this form. So because there aren't new things like in the juvenile form where a bone is, those categories get much more broad. All right, to estimate biological sex, we look at the pelvis. So the pelvis is really the only functionally different part of the human skeleton between males and females, because obviously males are going to be shaped differently in order to let a baby pass safely through that pelvic space. So we look at a lot of characteristics that are reflecting that fact, reflecting the fact that females are able to give birth. And so we look at a lot of characteristics that see if I can draw here. We have female on the right, male here on the left. And as you can see, the female pelvis is much wider. So this is what we call the pelvic brim or the pelvic inlet here. And these would be your hip bones that you can feel at your waist up here. So the pelvic inlet is much wider in females, much more narrow in males. And that wider versus narrow is just going to be consistent through a lot of these features that we're looking at. So we'll look at this angle here, the subpubic angle, that contour. These are your ilium, and they're flared out more, creating this wide bowl shape. Whereas in males, they're much more vertical and narrow. This space of the pelvic inlet is more narrow. The angle is...
While you're reconnecting, let me just say the CA pound Institute there is named for C. Addison Pound, who married into the Baird family who owned Baird Hardware. And so he had some money to give, which probably is the result of that. But he also gave the land for the current Gainesville High School on Northwest 13th Street.
I did not know that.
I think, I think you were able to hear about the pelvis, so I'll keep on moving. But as I was saying, a lot of the changes that we're looking for is just because it's the functionally different part of the skeleton. Everything else, there is a functional difference between male and female. So as of course, as I'm sure you all can guess, nature really isn't this cut and dry. There isn't a male box and a female box and everything kind of fits nicely into them. Biology exists more on a spectrum. So there are very robust female skeletons and very gracile or smaller male skeletons that kind of go against that trend you would expect. There are, 6 tall bodybuilding women and naturally very slender or shorter men. So we look at these traits on the skeleton on a spectrum of expression, and we grade them on a scale. So what you're looking at here on the right is half of a pelvis. So the pelvis is made up of multiple bones, and this is one of your os coxa, and it's just highlighting this notch right here called the greater sciatic notch. And then this diagram next to it is showing 1 to 5. What are the expressions of that notch? So we look at this to estimate sex in a pelvis. The scale of 1 to 5 is 1, what we've labeled the hyperfeminine state, and 5, what we've labeled the skeletally hypermasculine presentation. So most often you'll find females presenting with a 1, 2, 3, and males presenting around the range of a 3, 4, 5. But like I was just talking about, there's this middle ambiguous zone where biology isn't a binary. It isn't just a clean male female box or black or white. It's more complicated. It really is a spectrum that we see. So you can have more robust females presenting as a 4 in all these features of towards the more masculine side, and you can have male individuals presenting more towards a 2 on this spectrum. So the pelvis is the most accurate way to assess the skeleton, assess sex, on a skeleton because of childbirth. But we also look at overall size of the joints, where again, males tend to be larger, females tend to be on the smaller end of the spectrum. And we can look at certain features on the skull as well. So these are just diagrams on the left here looking at certain features, like this row is the mastoid process, this feature below the ear, or the thickness of the orbital rim here above the eyes. So we can look at certain features on the skull again, just looking for larger muscle attachments in men, smaller muscle attachments in women, and grading them, gauging them on that scale. So this is just from another perspective. This first row here, we're looking at the feature of the external occipital protuberance, which is this bump on the back of your head here. So men tend to have larger neck muscle attachment sites on the back of their skulls. So they usually present with a 3, 4, 5 here, where you can see this is the back of someone's skull, and you can see that bump getting bigger as you get more and more towards that 5, the hypermasculine state. You usually only see this characteristic on male skeletons. So we'll look at features in the pelvis, measure the joint sites, and we'll look at features on the skull as well. All right, and that third component of the bioprofile is self-explanatory stature. We're just measuring elements of the skeleton that are that estimate living height of a person most accurately, and then we produce a stature range for investigators to compare to medical records, a driver's license, anything like that of a missing person that you could see if they're within our stature range, and if they might be that individual. And then the fourth component of estimating ancestry is much more complicated, because notice I'm not using the word race. which isn't really a term based in biology, it's a social construct. So we've created these broad categories like Black, white, Hispanic that we use conversationally in life, but they don't have biological meaning. So humans are really much more similar than we are different. And at the same time, those terms that we use aren't capturing the variation and the diversity that exists within humans. So right now, we can estimate in forensic anthropology that someone was Hispanic or Black or white, but what do those terms really mean? Does that mean their ancestors were from Haiti or Ghana or Zimbabwe or Guatemala or Mexico? Estimating someone's ancestry or population affinity, which just means how well do they map into a certain population. This is really undergoing a lot of change in our field right now and could be a much longer conversation. So for today, I'm just going to show you how we statistically do this in the job right now. So we use a database called 4DISC that houses skulls. So it's just a large database where we've collected data on skulls that we know the ancestry of the donor. And then we measure the same places and the same distances on an unidentified skull, on an unknown skull, and then put them into this database to see how well that skull, that unknown skull, which group does it map closest to? Is it an example of it here? So this is what it looks like. You're inputting those measurements into the system, and then it gives you a report like this that tells you which group does your skull classify closest to. So there are a limited number of groups, so we need to really build that database to show the complexity of humanity. But as you can see, this skull is plotting closest to white males, WM, but it's not a very strong classification. So we can look at the graph form to make it a little bit more clearer. So this just allows you to visualize your results of this X represents our unidentified skull here. And you can see there's a lot of overlap between these groups of white male here, Black males, Hispanic males. So this would be the Black male circle, Hispanic males. There's a lot of overlap between the group. Like I said, we're more similar than we are different. And there isn't one skeletal trait that I could look at a skeleton and say, okay, this has to be this ancestral group, because we all share the same skeletal traits. So what we're really providing is this percentage probability that it's falling within that group. So if our skull had fallen out here in the white male circle, I'd feel we'd give a higher percentage probability. I'd feel a lot more comfortable saying that that was a white male. But this doesn't really give any indication of what that person looked like or how they identified. So assessing ancestry is really in its infancy as a science. And that kind of concludes the bioprofile. So now we can move on to just other things you can learn from a skeleton. So we'll start with pathology. So we also look for any we want to give the most complete report as possible. So we do that bioprofile, but then we also look at what diseases were possibly on the bones that could help us ID that victim. So if they can be in certain categories. So we look, is it infectious, like osteomyelitis, that you see in the picture on the right? Is it degenerative, like osteoporosis or osteoarthritis, which you see in this middle photo here on a vertebra, which basically everyone acquires as we age. You know, our bodies carry us around for decades, so that shows in our bones. But it can also be a congenital pathology where sometimes you are born with something distinct enough to identify you. And then another important thing you can look for in the skeleton is evidence of trauma. So we're able to categorize trauma as either antemortem, perimortem, or postmortem trauma marks to the bone. So antemortem, ante meaning before, mortem meaning death. This refers to everything that affects the skeleton while you're alive. So bone is a living system. If you break a bone during life, it's going to try to heal itself. And there's going to be evidence of that healing on the bone, which we call a callus. And you see that here. So in this picture, this is again your femur, largest bone in your body. So this is a pretty significant break. But you see this callus right here where the bone was broken and then healed itself in this little kind of cocoon there that restabilizes that area. And the person obviously lived long enough for this healing process to occur. So if somebody's broken a bone, we're going to see evidence of that in their skeleton. And then the next category is perimortem. So perimortem is damage that's happened to the skeleton around the time of death. And we can tell that that break happened around that time, around the time of death, because the bone hasn't had any time to heal. There's no evidence of healing. And we can see a picture of perimortem trauma over here on the right. This break of the femur is very sharp, very jagged, no healing has occurred. And that fracture site is the same color as the rest of the bone. So the bone's been soil stained brown. And if that break had happened 5 years after the person died, an elephant steps on the bones, that fracture site would be much lighter in color because the outside would have been stained and then it would have fractured. So the fact that it's the same color tells us it happened at the same time that the bones were being stained. So any trauma like gunshot wounds, knife wounds, all that is going to be classified as perimortem trauma. And then that last category, postmortem, is referring to all the changes that happen after death. So we also call this taphonomy. So this is one of my favorite things to assess in a skeleton. Taphonomy, everything that happens to the skeleton after death. So how are the bones interacting with the environment? So these skulls are actually giving us a lot of information if we look closely. Both of them are showing differential preservation. So what that means is part of the skull was in one environment and the other part of the skull was in another environment. So this brown staining on the picture on the left, you can see brown staining on the top of the skull. That indicates that the skull was upside down in a soil environment and it's been stained brown by that soil. But then you can see this portion of the skull is a dark green. So that portion was above the soil level, but stained by algae or really rich plant matter. And you can see, so it gives you an idea of reconstructing that environment and reconstructing what's happened to that individual after they died. And then the picture on the right, same idea, the skull itself is bleached white. So that tells me that it was above the surface. It wasn't buried. The sun has bleached it white, but the mandible here has been stained a brown. So that tells me it was buried in the soil in a different environment than the skull. And you can also see root etching where roots of plants nearby wrapped itself around that mandible. Other things we can look at are how animals have interacted with the bones. So birds like vultures or eagles, they're going to leave different marks than, say, a rodent or larger carnivores like a wolf or alligators in Florida. So the signature left on the bones that we see back at the lab, like this picture here on the right, it's going to be really reflecting two things: the type of tool that made that mark, so a wolf tooth, a rodent tooth, or a knife, and the purpose of that tool. So where on the skeleton were those cut marks? Were they made by wolves trying to get at tissue or bone marrow? Were they rodents who just come to dry bones to file down their teeth and get minerals? Or were they made by humans? That could tell us something about cause of death or intention of a human. And of course, bones can be damaged by fire, either intentionally or unintentionally. And you can see the spectrum of fire damage here on the picture on the left, where you really have a spectrum of color depending on how long the bones are in the fire or how hot the fire was. If the fire is very hot, you're going to see that color change much quicker than if it's a slower burning fire. And on the right, you see water damage. So you can track barnacle growth and also in different environments, you really can reconstruct what type of water environment the bones were in. If they were in a whitewater rapids river, bumping against rocks and smoothing down their edges, they're going to look very different than bones that were in water in a still standing lake that have just settled down to the bottom and maybe have more, say, barnacle activity or plant or animal activity. So we can reconstruct a lot about the story of what's happened to somebody by looking at their skeleton and looking at how those bones have interacted with their environment. And I'm ending the presentation today with a couple examples of real world applications of what forensic anthropologists do. So these are just a couple projects that forensic anthropologists have recently been working on in the world. The first being an excavation that I worked on last year. So this is the Oklahoma 1921 Race Massacre Excavation Project. the University of Florida, and my supervisor, Dr. Phoebe Stubblefield at the Forensic Lab, are working with the Oklahoma government to excavate this cemetery plot. So just a brief backstory. In 1921, there was a massacre of the Black residents of Tulsa, Oklahoma by a white mob. And after the massacre, their remains were buried very quickly after the attack. But on the 100th anniversary, the city of Tulsa is trying to re-examine that history and learn more about it. So those skeletons are being exhumed, and by studying their remains and studying the trauma and listening to the families of those victims, we can learn more about what really happened at that event. And that is a lot of what forensic anthropologists do. You learn what you can from the bones to reconstruct history and be able to take a few of those last clues that remain about historic events and be able to tell that story again. So here we are, just assisting the state of Oklahoma in locating the remains and finding that balance, like I talked about before, between very careful archaeological excavation and a speedy forensic recovery. And the final example is the Defense POW MIA Accounting Agency. So this is the largest forensic anthropology lab definitely in the country, maybe the world. It's located in Hawaii, and it's a military operation trying to recover the remains of US soldiers that died either missing in action or as prisoners of war, roughly in the last 100 years of US military conflict. So they travel back to places like Laos, Cambodia, Vietnam, and recover skeletal remains of soldiers, identify them if possible, and then return the remains to their families. So it's that type of humanitarian work that I really hope to do in my career, as well as working at the state level like I do now with medical examiners and law enforcement. So. And that will be the end of my talk. So if you have any questions, I'm happy to answer them.
I have one question, and there may not be an answer for this, but we've been hearing a lot over probably the past 5 years or so about trans people, people who are one sex biologically but identify as another. And I'm just wondering, since that seems to be something that's almost born in the person, whether their body, for example, if a woman is identifying as a feeling as a man, would you expect her, those categories 1 to 5, that maybe her body might have gone more toward the 4 end? You know what I mean? Could you just speculate about that?
Yeah, I'd be happy to. That's a fantastic question. I was actually just talking to my aunt about that last night as I was practicing this presentation. So I think a lot of people are curious about this. So It really depends on at what time in their life that the person decides to transition and what sort of transition they're choosing. So some people, when they're transitioning through a gender, have gender reassignment surgery. And so you might have feminizing or masculinizing surgeries to change the structure of, say, a sharp jawbone or the nose. And so we'd see evidence of that, of those surgeries on the skull, and you might be able to use that as a clue. But if that's if the person has had, not everyone has surgeries when they're transitioning. So that's if they've had surgeries later in life. There's actually, like you're talking about, this really is, in the forensic anthropology field, a new thing that we're considering, and rightly so, because you don't want to erase, trans individuals when you're trying to do forensic identification. So you can also have hormone replacement therapy. So if the hormone replacement therapy starts before puberty, what we see in the skeleton could be different than the biological sex that they were born with. So I guess it really is speculation, like you were saying. These studies are just happening right now. Andrea and I have a friend in our lab at the University of Florida who's studying this for their PhD. So the question is, how do you make sure that you're including trans individuals in your forensic identification? Are you looking for those signs? Do you know what to look for? But yeah, you would, if they haven't had hormone replacement therapy, if they haven't had any surgeries, then it is a danger that you would miscalculate and misestimate their biological sex. So you really have to be considering that as you're doing your, consider your own biases and consider that as you're doing your forensic identification.
Thank you.
Yeah.
Ken Burns, go ahead.
Yes. One thing you didn't talk about was the ability of DNA analysis to complement your study of the bones or the skeleton. Can you make any comments about that?
Sure. Yeah, there is a national database that we use called NAMUS that multiple forensic anthropology labs around the country use, where you get skeletal remains and you take a sample and send them to that lab, so that it's a, if somebody crosses a state boundary, you're not only working with state databases and lose the ability to identify them. So we send in DNA samples so that you can compare across the whole country. The problem is the only DNA that goes into that system are victims, and sometimes if you if you are sent to jail, sometimes criminals will also get their DNA put into a database. So families of victims often submit DNA as well to compare to the victim database. But yeah, DNA is definitely a tool we use for positive identifications. We also include other things that can help with positive IDs. Are if you've had a surgery and there's a surgical implant, those have serial numbers, unique serial numbers. So that can be tracked down to the individual, to the hospital, to the unique distributor for that part. So there are a few other techniques like that that allow more specific, positive identification for an individual.
I think they also allow you to I don't know what you'll call it, ethnicity, but you should be able, as Ancestry.com claims to give a pretty good idea of the background of the individual.
Mm-hmm. Yeah, the few issues with that are just DNA is really expensive to run. So it often it's not routine, I guess. If the money is there, then it gets run, but it can be thousands and thousands of dollars, which often isn't really feasible for local law enforcement to run that, to just kind of get a a rundown on every person. But at the same time, we can know someone, like you're saying, ancestry.com, we can know their very specific ancestry, but that also doesn't tell you exactly what they look like, since there's such human variation. And it also doesn't tell you how they might have identified, but it definitely, DNA is definitely helpful.
So the last sort of comment or thought is that when you were talking about cranial measurements in terms of ethnicity, it started to sound like the eugenics movement of the 1930s, which was not a very desirable biological study. Let's put it that way.
Agreed. Yeah, we are trying to steer as far away from phrenology and eugenics as humanly possible. Yeah, so the main difference is within phrenology and controlling through eugenics, we're not making comments on ability of, phrenology was looking at the skull 100 years ago and saying, oh, because of this bump on your skull, you're a criminal or you're less intelligent. And that just happened to coincide with a lot of white supremacist thoughts of magically white skulls were not showing criminal signatures or a lack of intelligence. So yeah, I agree. It's that same kind of language, which really requires scientists to be considering all of their biases and really know our history so that we stay on the right side of ethics. But then also notice that we're not making comments about if that, that isn't in any way linked to intelligence or morality or anything like that. Yeah, but yeah, it is an interesting thing to keep in your head for sure.
Walter, you were next.
Yeah, Ken started my question about DNA, but I would like to follow up with where in the skeletal remains are the best places to extract DNA, and what if you get an incomplete DNA sample, can you learn from that, or do you have to have a complete run of the genome?
This isn't my area of expertise, but I can tell you what we do in our lab. We have a geneticist, a chemist, Sam McCrane in our lab, who usually, who is the specialist on this. But so you can take DNA samples. Sometimes you take what you can get of not the skeleton, the full skeleton isn't always there. So what you have is what you run, but a lot of samples are taken just from the mid shaft of your long bones. So the mid shaft of your humerus or your femur. You can also use teeth because the teeth are made of a harder substance. So they're going to survive longer than the bones. So sometimes that may be the only thing left, and you can use teeth for DNA. Then you have to consider some of the taphonomic effects, like I talked about before, like fire burning. Sometimes if the remains are damaged, it's going to damage that DNA and you won't be able to run it. But typically you can take samples from the ribs. I think ribs and long bone shafts are what I've seen most often. You just want to avoid any of the specific sites that we look for: sex, age, ancestry, that sort of thing. So we don't really use mid shaft of long bones for any sort of assessment. So, and it's also a robust place, so they take it from there.
So, and typically you would not even run DNA if you know this was a person, the person, the name and everything. Now you're just looking for a cause of death, so that wouldn't require DNA.
Yeah, if there's already been a positive ID, then and they just want maybe a trauma analysis, you wouldn't need to run DNA.
All right, thank you. Barb, go ahead.
No, my primary question was already asked twice, but I also wanted to know about, I realize it's probably quite expensive and it's probably not your field, but how useful is facial reconstruction using muscle attachment sites for for largeness of muscles and then the bones underneath, etc.?
We don't have a person who specializes in facial reconstruction at our lab. I went to work at a lab this summer for a month in San Marcos, Texas. They have a forensic lab there, and they teach workshops on facial reconstruction. And it really, as I understand it, it's this balance between art and science, where you take the science of using, a model of a head and clay, and understanding what the thickness is, as I'm sure you already know, thickness of the skin, thickness of the muscles of the face, and estimating that biology, and then meeting it with artistic skill and artistic license of what you think, that person's facial structure was. I really don't know too much about it because we don't use it in our lab, but I certainly don't have the artistic skills to do it.
Yeah, the muscle attachment sites are very crucial in that, size and angles and so forth. Yeah, thank you.
Yeah, it is really interesting because a lot of the material that doesn't preserve, like the cartilage of the nose, or I guess like you have a little fat pad here, as I know, as an anatomist. But the just subtle changes in people's anatomy can change the appearance of your face so much that it seems like a really difficult thing to do. But I do know it's used around the country. Yeah.
Any questions here in the Oak Room? Layla, go ahead.
Hi, very interesting presentation, thank
Oh,
you.
thank you.
Do you all work with the University of South Florida? Because that was the only place I could donate my body for forensic anthropology, even though I'm closer to Gainesville.
Yeah, we actually just two-part question there. We actually just began a body donation program at the Cap Hill at the CA Pound Human ID Lab. So and it's brand new, so I'm sure whenever you asked about it, they didn't know. But yeah, we just started a body donation program at Cap Hill so people can donate their remains. But I've worked with South Florida just once on the I believe Dr. Walsh Haney works there, and she was working with us on the Oklahoma project. So I haven't gone down to visit their lab yet, but we do. It's a small world of forensic anthropology, so we definitely work with them a little bit. Yeah.
And can you address, when I had to fill out the application, there were some really specific questions as to illnesses and stuff like that, that they did not want a body that had certain illnesses. Can you address that?
Sure. Yeah, I can. I can guess is my only answer. So some of it has to do with safety of the technicians. Of you want to make sure that the people who are processing the remains are safe from pathological conditions, like we talked about. And I think that the lab that I was working at in Texas, I think they kind of have the same rules across the board of certain conditions they don't accept remains from. But I'm not sure if that's across the country. But that's really, yeah, just for the safety of the technicians who are processing remains.
This has been amazing. I don't see any other questions. Walter, Andreana?
Yes, thank you very much. I have a question not related to this talk, but related to all the talks. And I'm very thankful that Andrea has found these really interesting people. So I'm going to ask Andrea and ask her to ask her presenters, will you help me again next year? Because you've done a wonderful job. Thank you so much.
Thank you so much, Walter. Yeah, I'm happy to continue looking for more speakers. There's a few people I had like on deck for next summer, so I think we can definitely keep this going. Yeah, no, this has been great.
And will you tell us about next week's speaker? That will be our last speaker, I believe.
Mhm.
Yes. Our last speaker next week, we'll be hearing from Inez Said, who was in the Digital Humanities Institute at UF, and she recently completed the master's program there. And now she works full-time there as a software engineer. So she's going to be talking next week about her projects with virtual reality and augmented reality, super cool stuff, and how she hopes to make these technologies more accessible and easy to use. So yeah, we'll be hearing more about her projects.
So thank you,
sounds great.
everyone, for showing up. I'm showing up from home again, as you can see. But I really have enjoyed all of the talks so far and look forward to the talk next week and look forward to the time we could all get together and actually see each other without masks and talk about what you've done and to thank you for all the work.
Thank you so much. And thank you so much again for being here today, Rose. This was great.
Thank you,
Yeah, thank you
Rose.
for having me.
Enjoy Alaska for us. We're sweating
Thank
it
you, I will.
out. Stay well, everyone. See you again.